number
3.
1, 2, 3.
Okay, I'm Morgan, and this is Jennifer and Connor.
All right. And so let's talk about the first question. So why do you see a tubular band in the W and S lanes, but not in the P lanes? And let me just point that out what we're talking about here. So we're looking at the tubulin, at the blots that were tested with tubulin antibodies, anti-tubulin antibodies, just to be precise. So we see tubulin present in the W lanes in both cases, in the S lanes in both cases, but not in the P lanes.
Okay, we said because we know the P lane just contains a nuclei, so we know that there's no tubulin in the nuclei. So we know there has to be tubulin somewhere in the cell, just somewhere outside the nuclei.
Okay, so in the cytoplasm, so this is telling us that tubulin is present in the cell. It is present in the cytoplasm of the cell, but not in the nucleus of the cell, in both wild-type cells and mutant cells. How about another row in seat number?
Row 4, seat 4.
4, seat 4. And let's go on this side. 1, 2, 3, 4. 1, 2, 3, 4. John?
Joe.
Joe. Close.
Yeah. That's Con. That's Lauren. And that's Laura.
All right. So let's go to question 2. Why do you see an H3 band in the W and P lanes but not S lanes?
Because it's a nuclear protein, so it's in the nucleus. So it must have been centrifuged into the
And why do you see it in the W lane?
Well, simply because it's part of the cell. Yeah.
Because the whole cell has a nucleus, so of course it's there, right? Excellent.
So do you think that was a stupid experiment? Like, why do that, or is this your next question you're going to ask somebody else?
Let's
Okay,
do that. I was not, but
You can
so
go
I'm
ahead
going
and
to be
see while
doing
I-
5, 5.
5, 5. Okay, 5,
1,
5.
2, 3, 4, 5. So it's 1, 2, 3, 4, 5. Winner.
Hi, that's Maddie. This is Lou.
And your
So
name?
is that, what's the... what-
Hold on.
that
shh.
was-
And your name?
Sorry.
Paul. All right. So now,
Oh, was that a stupid experiment? I mean, we already know that histones are in the nucleus, duh.
It's always nice to confirm something, right?
That's right. And we call that a control. You're absolutely right. It's one of the most crucial things. It's not only just nice, it's actually crucial in science. So you centrifuged it, but what if you messed up the experiment and the nuclei were all over the place, or you didn't lyse the cells, didn't lyse them properly or something like that? You want to make sure you're looking at what you're looking. You're exactly right.
yeah. So we're using tubulin and histones as markers to tell us that we have done the separation of the nucleus and the cytoplasm correctly. Right. So this tells us that there's very little, if any at all, almost non-visible contamination of cytoplasm in the nuclei, and conversely that there is no contamination of nuclei in the cytoplasm fraction of the cell. So we did the experiment correctly. So these 2 lanes are simply serving as controls and tells us that we can now have the basis on which we can reach some conclusion about the unknown protein which is on top. All right? Okay, how about another row and number?
Is there 6, 3?
6, 3. Let's go on that side. 1, 2, 3, 4, 5, 6. 1, 2, 3.
Hi, I'm Josh. This is Hayden. What was the question?
So, okay, I haven't asked it, so let's go to the third one. So, in the case of RCC1, we see this result over here. What does this tell you?
It tells us that the RCC1 protein is found in the nucleus as opposed to the mutant, which it isn't found.
Okay. And anything else?
There was one thing that Hayden noticed that in the P and the S lanes of the wild type, you see it's a lot lighter than the S lane in the mutant. So we thought that it might possibly be because there's like a transport protein that's missing that isn't allowing the protein to be expressed thing in the supernatant?
Are you talking about the intensity of this band here, for
Yes.
example?
Yes.
All right, so possibly. So you guys are trying to do a little bit more quantitative analysis, and that's awesome. All right,
I disagree.
but that's where you need to begin to look at your controls. All right. So if you look at your controls, you can see also that your sample control here is more intense than the other ones. So you have to begin to standardize against some of your controls. All right.
Yeah,
So
but
this-
I think also what you said, if I understood you right, is that you're concluding because this is darker than this, that maybe the supernatant lanes, that it has something to do with the RCC1 expression. Was that your conclusion?
A
Did I,
little bit, or that the protein's expressed, but it's not
as much, right?
transported within the nucleus.
It's not transported into the nucleus.
That's right.
Correct.
Yeah, so, but as far as expression levels go, if we compare these two, this is roughly the same. And if we add up those two and sort of mentally add them, we can see that. So it looks like expression levels and the level of the protein are roughly the same. Yeah.
And
And I
of
may
course,
have misunderstood
being
what you were
quantitative is
saying.
really important, and you would have to do a little bit more detailed experiments to then be able to quantify, right? So you can see from this experiment that the wild type protein is present both in this nucleus and in the cytoplasm of the cell, but the mutant protein is present only in the cytoplasm of the cell. So what conclusion may we reach from that evidence? Give me a number.
1-1.
1.
This is Bella, Treasure, and Christoph.
And your name?
Teresa.
Teresa.
And I think that we concluded that the mutation might be that it's no longer located in the nucleus, so the mutation caused it that it wasn't able to be in the nucleus anymore, so it's all in the cytoplasm.
Okay. So, beautiful. So it's a mutation. This might be one interpretation. We need to do many more experiments to really nail it down. But this would suggest that mutation in RCC1 impairs its ability to enter the nucleus, to go into the nucleus, right? Somebody was asking, how is it possible that the protein is present in both cytoplasm and the nucleus? So if this is a nuclear protein, why is it not all in the nucleus like histone H3? Anybody has a possible reason? There's many possible reasons.
Could it be that proteins are sometimes created in the nucleus and then transported?
Okay, so maybe it's just transported sometimes. Is that what you're saying? Yep. So maybe it's upon a certain signal. Another reason is that this protein might be shuttling back and forth between the nucleus and the cytoplasm, going in and out, in which case you might have a balance of the protein in both places. There's many possible reasons that we cannot discern from this experiment. Right? But the fact is that this is a fact. RCC1 is present both in the cytoplasm and the nucleus in a normal state, but when you introduce this particular mutation, which we don't know what it is, then the protein cannot be in the nucleus. Boom, all right? So you've learned something new about RCC1 by a very simple experiment of breaking cells open, separating nuclei from cytoplasm, and doing Western blots with these antibodies. All right? So that's the power of such a simple experiment in the lab. Questions? Okay. So separating like that is nice and powerful, but sometimes we want to have more separation. So I compare the separation of components by centrifugation as if you were going into a big dumpster, which is the whole cell lysate, and you just came in with this huge crane. Oh, it's not gonna play. All right. No, never mind. Okay, fine. if you were to come in with just this gigantic crane and just grab the biggest things you can possibly grab. Sometimes you want to be a lot more specific than that. You want to go for a specific component of the cell that might be equally sized to hundreds of other things in the cell. So centrifugation is not going to make the trick. So that's where we can use antibodies in a different way. Remember, antibodies are proteins that have a specific binding pocket that binds tightly by non-covalent interactions with its target, the protein that it was made against, the antigen, right? So we can, essentially, we have tweezers that allows us to go into a complex mixture of proteins, grab the protein we want with our antibodies to pull it out. That experiment is what we call immunoprecipitation, which is, let's see if this plays, maybe not. Looks like all my movies are not gonna play. Oh, no, I see, media not found, I should have updated this. So we're gonna go with that movies. Did you have a chance to see it online? Yes? All right. So there's a link online to see that, so I recommend that you see it. So the idea is that you have your mixture of proteins that can be quite complex. So you have hundreds, thousands of different types of proteins, and you want to separate a specific protein or protein complex out of them. When I say a protein would be an individual protein, if I say a protein complex, that means multiple proteins bound together to each other in a specific way. Right? So what I can do is I can take little plastic beads or little beads made of some other kind of polymer, like agarose or polyacrylamide, that are big and heavy compared to a protein, and therefore are sedimentable in the centrifuge. And I can attach my antibodies physically, covalently, on the surface of that bead. So now I have beads that have hundreds or thousands of antibodies arrayed on the surface of that bead. So if I put in those beads in my test tube with my complex of proteins, the antibodies are going to be, as they're floating around, they're going to be able to bind and capture the protein antigen that they were made against. So now you have your proteins attached to an antibody attached to a bead. So I can do a very low centrifugation of this complex mix, and that's going to leave me with the beads. Only in solution, I will have only the beads with the antibody with the protein they bound, their antigen. Everything else is going to be in this supernatant. I can do this multiple times to wash away any contaminant protein that it was not actually physically bound to the antibodies. And now I'm going to have a fairly pure suspension of my protein of interest attached to the antibody. I can separate that interaction between the antibody and the antigen. There's a variety of ways I can do that, so that's what we would call, say, elute. So elution would be separation of the antibody from the antigen protein. So now I have a solution of what we would call an immunoprecipitate. A precipitated complex that was precipitated by antibodies. And now I can analyze it. All right? Questions?